Hello, are you bio students? So today we are going to look at 8.3, the Calvin cycle, and we're going to focus on how the Calvin cycle was discovered and who discovered it and how the experiment came about. So before we do so, let's go back to the light independent reaction, which we can collectively call the Calvin cycle. And if we look at step 3 here, we see that ribulose biphosphate, or RUBP, is what you need to start the carbon fixation process, right? You need ribulose biphosphate and carbon dioxide to get that unstable 6-carbon molecule. Now, after ribulose biphosphate is formed into a 6-carbon molecule, we know that that 6-carbon molecule is unstable and it splits into GP, which is a 3-carbon molecule. And then we know that that GP then gets converted into TP, which is triose phosphate. And that's what's going to give rise to the carbohydrate or the sugar or the glucose that gets formed. But then that TP, the 5 molecules that are left, needs to regenerate back into ribulose biphosphate. So the key idea here is that you have a 5-carbon compound here, right, known as ribulose biphosphate. Then you have a 3-carbon compound here known as glycerate 3-phosphate. And then you have another 3-carbon compound here known as triose phosphate. So how is this sequence of carbon compounds discovered? And that's the genius of what the Calvin cycle and the discovery of the Calvin cycle talks about. So let's see. The light-independent reactions are also collectively known as the Calvin cycle. We know that. And it's named after the American chemist Melvin Calvin. And he is the one that completed his Lollipop experiment to come up with the Calvin cycle and the different steps of the Calvin cycle. So what did he do? He used this apparatus that kind of looked like a lollipop, hence the name, the Lollipop experiment. And it was actually genius, of course it was, right? And let's talk about what he did. So first I'm going to play this, and I'm actually going to play it in mute so I can go over the steps. All right, so first, radioactive carbon-14 is added to a lollipop apparatus right here containing green algae. So the green algae here, what is it doing? Well, it's light is applied to it, so photosynthesis starts. And if we pump in air and carbon dioxide and we shine light, then we know that this algae is going to be able to photosynthesize, right? Now, the idea behind injecting the carbon-14 is so that this carbon-14 gets incorporated into these organic compounds or glucose, right? Or the triose phosphate or the GP molecule or even the RUBP molecule, so that we can track where this carbon is going. So then after different time periods, Calvin would stop the experiment. And he would stop the experiment by injecting the algae with alcohol so that it would stop, so that the metabolism would stop, it wouldn't continue. And then dead algae samples were analyzed using chromatography, and it separated the different carbon compounds. So remember, we have a 5-carbon compound, we have a 3-GP carbon compound, and we have a 3-carbon triose phosphate compound. So these compounds, after they were separated, they were identified using audio radiography, which is an X-ray film exposure. And then they were compared with one another to kind of find out their order as they were generated within the Calvin cycle. Okay, so let's look at here what's happening. I'm going to play and stop. So here we see, let's start it again. Here we see carbon-14. Turn off the sound here. Carbon-14 being injected into the algae, right? So photosynthesis is going to happen. And then at different time periods, the alcohol was injected or combined with the algae, right? So that would stop the metabolism. So then you would take samples of the different algae samples at different time periods, and you're going to separate them via 2D chromatography. So now you have the different carbon samples, they're separated, and then actually the 2D chromatography is different because you separate into 2 dimensions like this. Okay, so now you have a whole bunch of different carbon compounds there. And you are going to expose them like you would a photograph with an X-ray film in order to see them. Okay, and there they are. So there you have the radioactive compounds, or the carbon-14. And this is what was shown through the course of time. So if you stopped the experiment by adding alcohol at 5 seconds, you would see this. If you would stop it after 15 seconds, you would see this. And if you would stop it after 30 seconds, you would see this. So then what does that mean? Notice that this carbon compound here was identified as RUBP, the 5-carbon RUBP. So we see that after 5 seconds, you have a greater accumulation of this 5-carbon compound. After 15 seconds, you have a greater accumulation of GP, which is a 3-carbon compound. And after 30 seconds, you would have a greater accumulation of TP, this other 3-carbon compound. Okay? So based on that time frequency, Melvin Calvin was able to summarize the different steps of the Calvin cycle, right? So you would first start with RUBP, and then GP, and then TP, and then a regeneration of RUBP. So that's how it was timed through this Lollipop experiment, and that's how the different carbon compounds within the Calvin cycle were elucidated. So pretty smart guy, right? Yes, he was.